Coming up on Conversations on Bionics and Pain.
First of all, I just want to be clear and that no one misinterprets my position that, of course, I believe in plasticity. And I would never, ever claim that it does not exist, right? I think the only question is, what can and can't it do, right? And I think there is some naive ideas that it can do anything you want. And I think your study, is one of several that shows that it really cannot.
Okay, first of all, if the location of the sensation will have migrated, you agree that probably will be a great finding, and then the paper will have ended up in Nature or Science or something like that, but it didn't. And the reason why it will have been great is because the consequences of that will be that when building neuroprosthesis, it wouldn't really matter where in the nerves you put the electrode, the brain will take care of it later on. And it's rather disappointing that that doesn't happen. It just makes our work as designers a lot harder, because now we realize that we really need, to find new technologies to be able to selectively target those nerves or high-resolution interfaces that allow you to have projections to every part of the hand.
Yeah, but you know what? I mean, I just want to say a couple of things about that. One, the technologies that we're using now are pretty cool. I mean, you sent people home for years at a time with these technologies in their nerves, and they were very stable and they were useful, right? But they're still pretty primitive technologies. Right, It's very hard for me to believe that even in 10 years or 15 years, we're going to still rely on just a handful of channels, right?
Right.
And a lot of smart people are working on making those technologies better. So I think right now we're still at a stage where we're trying to get a lay of the land, try to understand what are the things that we want this technology to do. And then, have people who work on this kind of thing do that, right? I mean, we put a man on the moon. I think we can come up with a decent freaking, peripheral nerve array. But the other thing I want to say, to go back to the more basic science, in retrospect, it's obvious. It's obvious that plasticity, it shouldn't do that. Because first of all, if you think about, sensory systems have evolved, right? And they've evolved to kind of let you adapt to your environment. But if you think about it, an amputated animal is dead. There's no point in trying to come up with this really sophisticated way to reorganize the sensory system in order to, accommodate its recent injury, because it's not going to survive that injury. Right. That's just not something that evolution would actually work toward.
Correct.
Right. More generally, how do you construct a sensory system? Well, there's all this genetic kind of plan that is put into place, during development, that makes sure that, thalamic axons go to the right parts of the brain and just sets a scaffolding for, early kind of critical period learning. But once your body is fully grown, there's no reason for that. The sensory system, there's no reason for the sensory system to be plastic anymore. It's going to just tell you what's going on with your body, give you ground truth about the state of your body and its interactions with objects. So there's no reason for it to be plastic, right? And I think there's probably parts of the brain, like the motor system, there is a reason for the motor system to be plastic, because we constantly have to learn new skills and adapt to a slightly, to perturbations. But I just don't think that that's the case on the sensory side. And that's why I've been very skeptical of this from day one.
Welcome to Conversations on Bionics and Pain. I'm Dr. Max Ortiz-Catalan. I'm the director of the Center for Bionics and Pain Research, which is a multidisciplinary engineering and medical collaboration between Chalmers University of Technology, Sahlgrenska University Hospital, and the University of Gothenburg in Sweden. In this episode dedicated to a research article, I talked with Professor Sliman Bensmaia at the University of Chicago, with whom I collaborated on an article published in Cell Reports with the title of Chronic Use of a Sensitized Bionic Hand Does Not Remap the Sense of Touch. In this study, we provide evidence for the limits of brain plasticity with regards to somatosensation. In other words, our findings indicate some sort of neural hardwiring on the way we experience sensations such as touch. Whereas the motor system seems to be rather plastic or adaptable, trainable if you wish, The somatosensory system seems rather stubborn, unwilling to adapt to our engineering needs. We have known for decades that if we place an electrode in a nerve and send an electric pulse, the person will experience a sensation in the location where that nerve connects to its biological sensing organ. If the axon that is stimulated connects to a sensor in the index finger, you will feel a sensation in the index finger. Now, even if the index finger has been amputated, you will still experience a sensation in the index finger when stimulating that axon. And that is because your brain sees information arriving in that particular channel and infers that it must come from whatever it was originally connected to. So this is well known. Now, it is currently impossible to precisely place electrodes in nerve fibers that connect or used to connect to biological sensors of your choice. We know roughly where the sensations will be felt based on anatomical arrangements that tend to be similar between humans, but not precisely. If we place an electrode in the medial nerve in an above-elbow amputation, we know that sensations will be felt on the palmar side of the thumb, index, and middle finger, but not exactly in which one. And this is a problem because what you would like to do is to place electrodes in the parts of the nerves that precisely match the location that will be equivalent to where you have placed sensors in the prosthetic hand. So when you see the hand making contact with an object, that part making the contact is the same part that you feel the tactile sensations arising from. However, since this cannot be fixed at implantation, a naive hope that I had, and many others in the field, was that the brain will adapt and fix the mismatch after repeated exposure. Say, after thousands of times of seeing the contact in one of the fingers but feeling in another, the brain could rewire to match the visual feedback and the tactile experience. Unfortunately, we found out that this is not the case, and this is the topic of this episode. When writing the paper, we realized that there was really no scientific evidence to suggest that this will happen, and it was more wishful thinking or naive intuitions that led many people to believe that this will be the case. This is another example of why listening to one's intuitions is not a reliable way of arriving to the truth. Science does a much better work, and here I'm going to diverge a little bit on a public announcement. Do not follow your heart. I cringe every time I hear someone in pop culture saying, just follow your heart, because what they're basically saying is follow your intuitions. And how is it that intuitions arise? They arise by a mechanism that evolved millions of years ago for the survival of our species in a completely different environment than the one we live today. And therefore, this is no longer a reliable mechanism for decision-making or forming beliefs. There is a large body of literature arguing about this, and I'll be happy to touch on this in some other episode. But the point here is that the scientific method supported by reason and logic is a way much reliable way to arrive to the truth of a matter or decision-making for that end. And just to be clear, one can listen to one's intuitions if one is going to try to disprove them using evidence. And the best way to collect evidence is by using the scientific method. Now, you could expect that what's published in a scientific journal is closer to the truth, but this is not necessarily the case because the problems with science is that it's conducted by humans, and in humans we have a bunch of biases. And even if we do our best to compensate for those biases, there is always a lot of methodological problems when we are conducting experiments. Case in point, when I was running this experiment, we fitted a prosthetic system that allowed patients to perceive sensations coming from the prosthetic hand. The location where the patient perceived the sensation wasn't exactly the location where the sensor was placed. And again, the idea was that hopefully it will change over time. So we were asking the subject, where do you feel the location? Every other day, we're calling over the phone. And, subjects or participants in your experiments, they're very good at realizing what you want to hear. So there was, after a couple of weeks, the subject starts saying like, oh, well, actually, I think now I feel the sensation in the thumb. Where is the sensor located? And I got very excited, and I suppose that he perceived that I got very excited. And then he kept reporting that for a couple of times, but then he will be back on the hand. And then at some point, I felt like I needed to run some sort of control question. So I asked, so where do you feel the sensation? And he said, like, well, it's in the thumb. And then I said, oh, that's very strange because it shouldn't be moving to the thumb. And then he said, well, actually, no, it's on the palm and pretty much always in the palm. And you can't blame the guy because of this, because at the end, you spend a lot of times with these subjects, you helping them to restore some function, you provide them a prosthetic system, and they try to do the best to help you on your experiments as well. But this has consequences for people doing research in the field, because as of today, many of the experiments done with implanted electrodes is with, few subjects, a couple. Most of the papers are published with 1 or 2 or 3 subjects at most. So we have to make our best to try to account for biases and account for the subjects that participate in our experiments to try to place us, and that kind of deviate us from the truth that we're trying to find. In this case, we reported the findings of 3 subjects, but we have verified this with up to 6 subjects, and we can consistently see that the locations of the percepts are not changing. So now we're rather confident that this is the reality. But now we can jump into the discussion with Professor Bensmaia, and if you have any questions out of the topics that are addressed in this podcast or any other episode, you're welcome to reach out, and I might do an episode just for answering questions. Would you mind introducing yourself and speaking a little bit about how you end up being a professor?
Whoa, that is not quite the question you told me you were going to ask me. So my name is Sliman Bensmaia. I'm a professor in the Department of Organismal Biology and Anatomy at the University of Chicago. And I took a very circuitous route to becoming a professor. Just to make a long story short, I wanted to become a rocker. Things didn't really work out. And I just stumbled into a lab that studied the sense of touch as a graduate student, and became passionate about it and followed through with it. Learned to do neurophysiology. And then after that, learned that maybe this sort of basic scientific knowledge could be applied to, something useful like neuroprosthetics. And here I am.
Right. I mean, you've been doing a very fruitful work in this year. So I think it was good that you ended up in the field. So a few years ago, was it about 6 years ago, you were visiting Professor Johan Westberg in Gothenburg?
Something like that. It was a while ago.
Yeah, so that's when I got to meet you. Yep. And Johan introduced us, and we were in this conference room, and I was presenting the stuff that we were doing. And I said, well, we implanted this system, and the patient is having sensory feedback. It's not exactly in the location where it should be, but we're hopeful that it's going to switch. And then you were like, no, that's not going to switch. And then we set up a bet about a beer back then, where you said, like, it will not switch. And I was very optimistic that it will. And then it didn't. So that's the paper we published recently. But
my favorite part of that story is that I see you like many years later, like 5 or 6 years, however long. And you're like, Sliman, you won that bet. And I was like, what bet are you talking about? It's like, and then you told me the story, and I'd completely forgotten about it. And I don't think you ever gave me those beers, by the way.
No. So I remember it because, I wouldn't pass them free beer, but I also want to pay it. But, we haven't met since then, actually.
Right. Fair enough.
But after the Corona, then we'll settle that. Would you mind describing how is it that we experience sensations of touch?
Right. Well, that's a big, that's what I spend most of my time trying to figure out. And it would take a very long time to explain all of that. But in the very short of it is that you have mechanoreceptors embedded in your skin. And so what a mechanoreceptor is a little biological device that when mechanically stimulated, so when the skin deforms, it produces a neural signal that it sends then to the central nervous system. And we have basically 4 main touch receptors in the skin. And they differ in the way they respond to skin deformations. So some of them really like slow moving things when the skin deformation is slow, others like fast skin deformations, etc. But anyways, anytime you're interacting with an object, you have this pattern of skin deformations that then, activates, large numbers of these receptors. And the signals from these receptors are carried to the brain via these nerves, mainly the median and ulnar nerves for the palmar surface of the hand. And when you grasp an object, you have all this information about the object, its texture, its if it's moving across the skin, you have information about that, its shape, its local geometric features, and all this information is carried in these signals from these mechanoreceptors in the skin. And then downstream from there, you have very sophisticated neural circuitry that tries to extract information from these patterns of neural activation. And that's what we spend most of our time doing in my lab, is trying to understand, well, what is the pattern of neural activation in the nerve and at every subsequent stage of processing that conveys information about texture, or that conveys information about motion, or that conveys information about shape.
Right.
And I think an important thing that I would just want to mention, because maybe it's not obvious to people who haven't thought much about touch, and that is that, touch is a critical sensory modality for our ability to move our bodies, and especially the ability to do things with our hands. And anytime we're interacting with our hands, we rely very heavily on these signals from the skin. And without these signals, we struggle to do anything, and we continue to struggle to do anything, even with practice, because the visual feedback that you get when you're interacting with objects does not give you the information that you need to interact with them.
It's not enough, right? So very roughly, you'll say we have these biological sensors throughout the body in the skin. When they interact with the environment, they send signals up to the brain via the nerves. And then there is very sophisticated processing circuitry in the brain that makes experience of touch. And
Right.
So what we're trying to do in neuroprosthetics is restore sensations by interfacing with the nerves and then trying to simulate the activation of those biological sensors. So what we did in this experiment is that we have this technology that we call neuromusculoskeletal prostheses, where we can connect to the nerves, muscles, and skeletal of the patient to provide mechanical connection, but also electrical connections to the human nervous systems, the control system in the body, with the control system of the prosthesis. So we place an electrode in the first patient that we implanted is going to be about 8 years now, an electrode in the ulnar nerve that when we send electrical pulses, he reports experience of tactile sensations in the palm, in the area related to the ulnar innervation that will be close to the little and ringer finger. And because we have that electrode there, we can then have a sensor in the hand, and every time the hand touches an object, we detect that, and then we send pulses to this nerve, and then the patient experiences sensations. But the sensor in the hand is in the thumb, and he experiences sensations in the palm. And that is because it's very hard during the surgical procedure to be able to tell exactly which nerve you are connecting to. So you can connect to the ulnar nerve, but the ulnar nerve has a lot of axons, and those axons will innervate different areas in the hand. And it's very hard to tell exactly which area in the hand your electrode gets closer to. And
I mean, it's impossible, right? Basically, you have these fascicles in the nerve, and you're just sticking your, electrode on those fascicles, and you get what you get. There's really no a priori way to know. I mean, with existing technologies that I know of, at least,
right,
to know a priori where the sensation is going to be experienced when you stimulate from a given fascicle, right?
Right. So we have a rough idea of an area, but it's very hard to precisely say, okay, I want to be there. And then the hope I had, and I guess we had in my group in the beginning, was that because the patient will be using it in daily life, over time, the brain will compensate for the mismatch and then create the experience of having that sensation in the same location as the patient was observing touch to happen. But then these patients have used the system in daily life for a couple of years now, and that sensation has not moved at all. So in our paper, we show the report location, and you see a little bit of movement, but it's rather just random movement. And I will say that is basically coming down to the error of the measurement, because it's quite hard for someone to say in their phantom hand, this is exactly what I feel the sensation. So that's a couple of millimeters here and there in all directions. And I would say that's probably what we are seeing. And so the thing is that despite that the patients, and we wrote a paper about this, they see this arm as part of themselves. You can say it's embodied. They use it in functionally relevant activities. There was no change. And I guess so throughout these years, a lot of people thought that change will happen. Why do you think a lot of people naively, believe that?
Right. First of all, I just want to say that the experiment that you did was really I think the best test of this idea, right? That eventually you see this mismatch. So these subjects go home, use this prosthesis in everyday life. Every time they see their prosthetic thumb touching something, they feel something somewhere else on their hand. And you would think, right, eventually the nervous system would sort of kind of resolve that chronic conflict between what you see and what you feel. And eventually the felt experience would coincide with the visual experience. And what you showed is that it didn't, even after, like you said, just, this is functional use of this arm. So you're performing activities of daily living every day at home. You see this mismatch, there's this mismatch between what you see and what you feel. And yet the sensation never migrates to where it's seen, right? So I thought that was really an excellent test, because most people, people who were trying to do this kind of thing, were doing it in the lab, and so just for an hour at a time. And therefore, even if there were some plasticity there during, some movement in the lab, you would expect it to go back. It might just be pulled back in everyday life. Right. And so it was the right kind of experiment. And I was pretty confident it wasn't going to work. But if any experiment would have, I thought that one would, right? Just for the record. So, and as you and I were writing this paper, we started thinking, why would you even think that it would move, right?
Right.
Honestly, there's not a single thing in the literature that would lead you to believe that these percepts would move. And I think it's more like it's a combination of magical thinking where, and I'm not accusing you of magical thinking. There's another thing.
Which I would say is wishful thinking, yeah.
Okay, wishful thinking, all right, fine. Which is, the idea, there's this, I think, naive idea that the nervous system is really plastic and can learn anything. And I think this is, this paper is one of several papers, I think, that show that that's just simply not the case. But, there's also data that, if you don't think about it carefully, seem to suggest that this kind of plasticity exists. And I think the most compelling of those papers, or those studies rather, are studies of sort of what's been mislabeled reorganization after amputation, right? Where what happens is, and this has been documented now in both humans and monkeys, when you amputate, you have, an arm. So, there's a huge chunk of the brain that is devoted to representing the arm, right? And so anytime you're interacting with anything with your hand, there's a specific part of your brain that is activated by that, right? And what happens is, when you now take away the input by, losing that limb, that part of the brain in somatosensory cortex that is, that receives this touch input from the arm and hand, starts becoming activated by touching other parts of the body, right. Oftentimes the face, right? And so that's been interpreted as, wow, the brain, this is like, look at this amazing reorganization. This part that used to be an arm part of the brain is now a face part of the brain, right? And it's a really cool phenomenon. And the phenomenon itself, the phenomenon, to be clear, is if you look at in monkeys, right? If you deafferent, let's say you do something that is, as far as the central nervous system, the equivalent of amputating a monkey, and then you look at the arm representation of somatosensory cortex, you find that, of course, it no longer responds to that part, the arm, but it starts to respond to the face.
Right.
And the same thing is shown in human amputees. And furthermore, when you touch the face, you kind of, create a sensation that's projected to the arm, right? Which is actually, that latter bit, I think, is a clue that maybe it's not reorganization. But then there's this beautiful series of studies that started with John Koss and that was followed up by Neeraj Jain that really gets to the core of this and really sort of explains what's really going on. It's not this magical reorganization where suddenly arm cortex becomes hand cortex or face cortex. Rather, what happens is that you're taking away the main input to these neural structures. So for instance, you're deafferenting a large chunk of the cuneate nucleus, which is in the brain, a structure in the brainstem that receives input from the peripheral, from the nerve, right? It's in fact the first structure that receives the input from the nerve. Now, if you take away that input from the cuneate nucleus is connected with a nearby nucleus called the trigeminal nucleus, which is like the cuneate nucleus, except that it receives input from the skin of the face, right? So in you and me, there is some connections there. But now if you take the main connectivity away from the cuneate that came from the hand, what happens is that you start to strengthen the connections between the cuneate and the trigeminal nucleus. So these sort of connections that used to be sort of like modulatory connections, who knows what they were really for before, right? Suddenly become the primary drivers of cuneate activity because through this, a process of homeostatic plasticity. If you take a chunk of neurons and you take away their primary input, through this homeostasis is going to strengthen all the other inputs, right? That's just a thing that happens. So what happens here, this was shown by Neeraj Jain, is that now you have this pathway that goes from the face to the trigeminal nucleus, and then to the cuneate nucleus, to the somatosensory cortex. And so in these amputated monkeys, these sort of virtually amputated monkeys, if you now silence the cuneate nucleus, all that activity in the arm representation that is driven by face stimulation goes away. And he did a follow-up study showing that, between the hand representation and the face representation, there's a septum. There is like a cell. There's very few axons that cross from the face representation in somatosensory cortex to the hand representation. This is just in intact organisms. So if you were thinking all this is in the brain, this reorganization is happening in the brain, then what you would think is axons would start growing across the septum from the face to the arm representation, thereby sort of promoting this quote unquote reorganization. And guess what? That doesn't happen. There is, if you look at the deafferented side and the afferented side for the cortex, it's identical. There is still no axons that cross. Okay. So it seems like that there is no evidence for remapping. It's just simple homeostatic plasticity.
So there is clearly something called plasticity in the brain, right? So I found interesting when the paper came out and you tweet about it, there was a lot of bashing against plasticity because I think I read even a comment saying like, well, plasticity is not really a thing or something like that. And I felt like it's maybe a problem like many in philosophy where it comes down to semantics, right? So if you take the classic example where you say, if a tree falls in the forest and there is no person around, does it make a sound? And it really depends what you mean by sound. If you mean by sound, pressure waves, certainly it does. It doesn't require a person to be there to create that. But if you mean sound as the conscious experience of sound, then there needs to be a person there to have that experience. So it really depends on how you define things. And I feel like there is clearly something called plasticity in the brain. I mean, Ramon y Cajal in the beginning said like, well, there might be not such a thing, but then you have the discovery of long-term potentiation, the neurogenesis and so on. So there are certainly changes in the brain that you can define as plastic, but the extent at which those can take place, particularly on the sensory system, seem to me a little bit more limited than in the motor system, where I read some papers that you can take a single motor neuron and repurpose it for something else. it seems very difficult to do that on the sensory system.
Yeah, I mean, first of all, I just want to be clear and that no one misinterprets my position that, of course, I believe in plasticity, and I would never, ever claim that it does not exist, right? I think the only question is, what can and can't it do, right? And I think there is some naive ideas that it can do anything you want. And I think your study, is one of several that shows that it really cannot.
Okay, first of all, if the location of the sensation will have migrated, you agree that probably will be a great finding, and then the paper will have ended up in Nature or Science or something like that, but it didn't. And the reason why it will have been great is because the consequences of that will be that when building neuroprosthesis, it wouldn't really matter where in the nerves you put the electrode, the brain will take care of it later on. And it's rather disappointing that that doesn't happen. It just makes our work as designers a lot harder, because now we realize that we really need, to find new technologies to be able to selectively target those nerves or high resolution interfaces that allow you to have projections to every part of the hand.
Yeah, but you know what? I mean, I just want to say a couple of things about that. One, the technologies that we're using now are pretty cool. I mean, you send people home for years at a time with these technologies in their nerves, and they were very stable and they were useful, right? But they're still pretty primitive technologies. Right. It's very hard for me to believe that even in 10 years or 15 years, we're going to still rely on just a handful of channels, right?
Right.
And a lot of smart people are working on making those technologies better. So I think right now we're still at a stage where we're trying to get a lay of the land, try to understand what are the things that we want this technology to do. And then, have people who work on this kind of thing do that, right? I mean, we put a man on the moon. I think we can come up with a decent freaking, peripheral nerve array. But the other thing I want to say, to go back to the more basic science thing, is in retrospect, before we move into completely in the engineering side of things, in retrospect, it's obvious. It's obvious that plasticity shouldn't do that. It shouldn't do that. Because first of all, if you think about, sensory systems have evolved, right? And they've evolved to kind of let you adapt to your environment. But if you think about it, an amputated animal is dead. There's no point in trying to come up with this really sophisticated way to reorganize the sensory system in order to accommodate its recent injury, because it's not going to survive that injury, right? That's just not something that evolution would actually work toward, right? And I agree that with but more generally, how do you construct a sensory system? Well, there's all this genetic kind of plan that is put into place, during development, that makes sure that, thalamic axons go to the right parts of the brain, and just sets a scaffolding for, early kind of critical period learning. But once your body is fully grown, there's no reason for that. The sensory system, there's no reason for the sensory system to be plastic anymore. It's going to just tell you what's going on with your body, give you ground truth about the state of your body and its interactions with objects. So there's no reason for it to be plastic, right? And I think there's probably parts of the brain, like the motor system, there is a reason for the motor system to be plastic, because we constantly have to learn new skills and adapt to a slightly, to perturbations. But I just don't think that that's the case on the sensory side. And that's why I've been very skeptical of this from day one. Right.
I want to touch in a couple of maybe more detailed particularities of the study related to how this thing was measured. Because the way I have traditionally measured perception thresholds on the sensations is by using a single pulse. And I know all the other groups are using a train of pulses. And the decision of using a single pulse versus a train of pulse is completely arbitrary for both ends. I just saw a single pulse as something that requires less arbitrary decisions on, say, frequency and duration and so on. And then if you look at the paper, what we're reporting, those points are basically the perception thresholds. Where is the patient reporting the faintest or weakest sensation? Because what we know is that when they're using it in daily activities of the daily living, we stimulate, and the intensity of the sensation is graded based on the force applied to the sensor. And that has as a consequence that the area of the perception increases depending on the strength of the stimulation. So the sensation starts in one point, and those are the points that you see in the figure of the paper, but then they expand the stronger the stimulation is. And in one case, or two cases, in two of the patients at least, the extension of that area cover the location of the sensor, where I think you could say, okay, actually in daily life, when the patient grabs an object at the maximum force, the sensation will include the location of the thumb. But nevertheless, even with that, if you can say maybe a little bit of help to the system to say like, oh, this is kind of close, it's in the same nerve, it's relatively close. When you grab it strongly, you actually feel it there because it's the whole thumb that where you feel the sensation. Even in that case, the weakest percept was still in the original position, pointing to a rather stable location of those percepts over time, which I think is an important point of the paper. Will you agree?
Yeah. I mean, again, it depends on why you're saying you're helping it, right? You're helping it by having it, overlap sometimes, but you can
wishfully saying that, ignorantly saying that might, give it a nudge towards.
Right, exactly. And I'm just not sure that there's anything that could nudge it,
right.
And I think, it's an interesting idea. One thing that's worth probably saying is, your single pulse strategy is interesting. I think it's a good point that, when you're using pulse trains, then it's like what frequency, what duration, blah, blah, blah. It's, all that goes away when you're doing a single pulse. What's interesting too, though, is that, this has been shown, I think, for now decades, that, if you evoke a single spike in certain types of nerve fibers, like slowly adapting type 1, rapidly adapting, you evoke a percept, right? Mm. And so probably at threshold, and another thing that you mentioned, but I'll repeat, is that when your threshold, when the subjects were asked, what is the extent of the sensation at threshold, it was just like very punctate, right? It was just a little point.
Right.
And so probably at threshold, you're just stimulating a single nerve fiber, right? And as you turn up the juice, you start recruiting more and more nerve fibers within a fascicle. And that's why you sort of have this increase in the area over a range. It doesn't just increase,
which makes sense,
forever.
right?
Yeah,
physiologically makes sense.
That's exactly what you would expect. That's right. So again, though, you're saying, well, there's probably some hypotheses about how plasticity would operate, according to which this overlap would help, right? But since I don't believe in any of them, it's hard for me to say it would help, because there's nothing that would help. Because in fact, there's this pretty fixed mapping between the patterns of activity in the nerve, patterns of activity in the brain, and then the resulting sensory experience, right? Let me say one thing too that I think is relevant to this conversation, and that of course is that you can take you can take somebody who's been amputated for decades, right? They haven't had a sensation in their hand emanating from their hand in 20 years. And then you implant their median and or ulnar nerves with an electrode array. And then you electrically stimulate through that array. And poof, they get a sensation back that is experienced on the hand, on that hand that they know is not there. And it's a vivid tactile sensation that is experienced there. So that means, right, what does that imply? That this pathway that went from the hand to the brain, to the sensory experience of the hand, is still there, hasn't really changed much, at least insofar as it still results in that sensory experience once you electrically stimulate the nerve. So that I think also points toward the relative stability of kind of sensory representations in adulthood, at least.
Right. No, I guess I was just trying to be the devil's advocate, because in this conversation is you and I, and we're authors in the
I know. It's like an echo chamber.
We didn't invite anybody on the other side, which I have to say, actually, I wouldn't know who will be on the other side, because the response.
I can give you some suggestions later.
Okay. No, but so this was very interesting. And then, when we were writing this paper, I came across something that I don't remember if I told you, but I find very relevant to this finding. And that is that, so I work with surgeons here in Gothenburg. And there is a surgeon who, Gennaro Selvaggi, he does a lot of sex transfer surgery. So in a trans man, what you can do is that you do, let me get the term right, phalloplasty with nerve anastomosis, which basically means that you take a forearm flap, you take a piece of skin, and you build a penis out of it. And then what happens there is that, actually, let me share you my screen so you see what I'm talking about. And I'll link this paper to the podcast. So when you do this penis out of your own tissue, you end up with two nerves. So you have a lateral and a medial nerve. And what you do is you connect the clitoris nerve to the lateral branch, and then the genitofemoral nerve to the medial branch. And that means that half of the penis receives sensations that are perceived in the clitoris, and the other half are sensations perceived in the thigh. And when you are using your penis, you touch the entire of it, right? So you will say, if you have penis for several years, which is patients have, you will say that those sensations, if there is such a plasticity, will come together and will just feel in the clitoris or or now the penis. But that doesn't seem to happen. So when they need to do some revisions or something, and they need to anesthetize the penis, they will inject the anesthesia in both sides. And when you inject the lateral side, the patients will report pain in the clitoris. And when you inject it on the medial side, they will report pain in the thigh. And I think this is, using biological reconstruction. You connecting these nerves in your daily life, if you wish. This thing is connected to you. You use it every day. When you touch it, you touch both sides. The brain is not remap, is not making that, I don't know if you'll call it remap or just extreme plasticity at this point to create an experience that just derives entirely from the same point. And I think this, very much confirms what we've been talking about.
Very cool. That is very much in the same vein. I mean, exactly. That's what you would expect. So they did do this tracking in this study. They did track sort of where the sensory experience is projected. Huh.
Right.
Very cool.
Yeah. So there seems to be evidence, and so of course what I've been trying to do now is see if there is evidence that points to the contrary, because if we could find a way for that, what I'm calling now extreme plasticity to take place, it will make our job doing neuroprosthetics a lot easier. So it will be a good thing to find out. It doesn't, unfortunately, it doesn't seem that it's possible. Yet, it's good to remain open to possibilities.
Absolutely. I think it's important as a sort of a philosophical point. I think it's important to stay open to possibilities. And just because I can't think of any way or reason why, this kind of plasticity is possible, I'm open to that possibility. But I think it's also important, and I think our job as scientists is to take a position on things, be flexible and open-minded about it, but say, this is the way I think things work. Prove me otherwise. And I think in the plasticity realm, there was, again, I think a lot of magical thinking, this idea that, wow, anything can happen. And there are some very prominent members of the neuroscience community that to this day really at least write as if the nervous system can rewire in whatever way you want. If you systematically kind of create an input-output relationship, it can rewire in whatever way you want. And I think we need to re-examine that. And I think this study is one of the sort of nails in that kind of magical pixie dust plasticity story.
Right. No, I think
coffin to finish the plasticity coffin.
I think it's important to. So I'm not sure if we must take position as scientists, but I think it's important to voice your position, just so for yourself and others to be conscious that there will be bias because of your position, and at least being conscious, you can try to do something to contrarrest their effect. But
what I mean simply is to have a clearly stated hypothesis,
right,
that makes very specific predictions. Like I had a very specific predictions about the outcome of your study, right? And if I end up being wrong, then I, have to admit that I was wrong,
and
then I will, and I would owe you the beer that you owe me now.
Correct. No, I think
to put too fine a point on it.
Right. No, that's an important thing. It's because I think that's the main difference between science and pseudoscience, that you have ideas that should be testable and do specific predictions. And I think as long as you stick into that, you'll do well in science. And I'm saying this just because, again, the audience for this podcast is mostly, students or young scientists. So
right. I mean, that's part of why I want to bring that up. But I also want to say, there's another point to this, and that is, I don't have a vested interest in this, really, particularly in the role of plasticity or lack thereof. But when I was doing this, when I started doing neuroprosthetics work, actually, this was in the context of brain-machine interfaces. Every time I give a talk, because the other thing I want to mention that's very important is that my, sort of perspective on neuroprosthetics, sensory feedback in neuroprosthetics, whether it be for amputees or individuals with tetraplegia, is that, and to me it's a truism, but not everyone believes this, that you want to basically speak the language of the nervous system to the extent that you can match with your device the signals that the nervous system naturally uses to communicate it, to touch information. You're going to create sensations that are natural, that are intuitive, and that sort of support behavior better. I mean, right?
Ideally.
And ideally, obviously. And to the extent that you can do that, great.
Right.
And so I would give my talks and say, look, this is what we're trying to do. We're trying to speak the language of the nervous system. And people correctly would say, well, what about the fact that, now you've taken out this input and there's the potential for plasticity. And, and honestly, early on, I didn't really know the answer to that question. I was like, intuitively, it seemed like we were doing the right thing, but then it's like, well, boom. And so I teamed up with Tamar Macon, who basically studies amputees and the neural correlates of amputation. And, together we looked at the literature and just looked for evidence for this kind of, plasticity in the sensory system in a variety of different contexts. Mm-hmm. Hand, what do you call it when you sew a hand back on?
Reimplantation,
right. The context of amputation, the context of tetraplegia, anything we could find that might be relevant in that you just kind of changed, removed, or modified, the natural input. And in no case that we could see, was there a situation where there was the kind of reorganization that we would need as neuroprosthetists, right, to kind of overcome this problem that you were talking about earlier, that you don't really know where the projected fields are going to be. And so the point is, so this was a long-winded way to say that this is sort of a lens through which we can then interpret what has been misinterpreted. So for instance, this idea, why is it that when you touch the face of an amputee, you can create a sensation on that's experienced on the hand, right? In some cases. And from the context of that, what happens is through this homeostatic plasticity that we were talking about earlier, you're ectopically activating parts of the nervous system that have been deafferented. And that is interpreted downstream as if it's coming from the original source of that afferentation, right? So it all kind of fits together in a very clear picture. Right. And that's why I've been kind of pretty vocal about this hypothesis and challenging others to prove me wrong on it.
And I must say, I believe the evidence of these referred sensations, I don't think that has been studied thoroughly. It seems to me that a lot of it is anecdotal reports, which might be fine, but we don't really know the extent of which this happens. If it's like a minority of cases or like big part of cases and so on.
Yeah, I agree with that. Mm-hmm.
Great. So thanks a lot, Professor Bensmaia, for a conversation.
That was fun.
And we'll definitely settle that bet as soon as traveling is possible.
Hey, you know what? I got vaccinated. So maybe sooner rather than later, my friend, I'm going to come to Gothenburg and claim my beer from you.
Right. Let's do that. You can subscribe to this podcast in any of your preferred platforms, that is Spotify, Audible, iTunes, or whatever it is that you use to listen to podcasts. And thanks to Dr. Enzo Mastino for the jingles.